Hello everyone, welcome back. Today I'm here for this 12th part of this questionnaire, and in this part I will discuss some questions from plant physiology. Let's get started. So the question is, magnesium is an important component of, our options are chlorophyll, pheophytin, cytochrome or ferredoxin. So its right answer is chlorophyll. Chlorophyll has a tadpole shaped structure, which is a larva of frog. तो ऐसी इसकी shape होती है. It consists of a head and tail, and its head is made up of porphyrin ring, while its tail is made up of a phytol tail. That is made up of phytol, that is a long chain of hydrocarbon. Its atomic number is 12 and atomic weight is 24. It's found in the solid state at 20 degrees Celsius. So this is the structure of chlorophyll. It consists of two parts, that is this head portion and this lower portion that is called tail. This head is made up of this porphyrin ring, which is made by the four pyrrole rings joined with magnesium ion. Magnesium, which is present in the center, and these are joined with this, these four pyrrole rings joined with magnesium with the help of bonds, which are known as coordination bonds. So there are four pyrrole rings and the head of this chlorophyll, and this position decides the difference between chlorophyll A and B. In case of chlorophyll A, there is the functional group methyl group, that is CH3, and in case of chlorophyll B, there is aldehyde group, that is CH2 group. So this is the difference in this position. So magnesium is an important component of chlorophyll. Next comes cytochrome and phytochrome. So these are again two important pigment systems. Cytochrome is related with the phosphorylation in an electron carrier, while phytochrome is involved in morphogenetic responses and found in all the plants. Cytochrome carries 2 electrons during oxidative phosphorylation and photophosphorylation. It is also associated with a number of other processes like respiration, nitrate reduction, while phytochrome performs morphogenesis, differentiation and seed germination. And this is the only biological pigment which exists in two forms, that is PR form and PFR form. This PFR form is the active form of this pigment, while this is an active form. If we talk about its chemical constitution, so the cytochrome is cyclic tetrapyrrolic compound containing iron, while this phytochrome is a linear tetrapyrrole compound associated with protein. Then finally comes this ferredoxin. So this ferredoxin is very important component of electron transport system, as in case of light reaction. So light reaction में जो photo system 1 है उससे electron receive करता है, that is ferredoxin, and reduces this NADP to NADPH. So this ferredoxin is electron carrier in the light independent reaction, which accept electron from photosystem 1 and become reduced ferredoxin before reducing NADP positive in the reaction catalyzed by ferredoxin NADP reductase. So this enzyme, so the culmination of ETC is the reduction of NADP positive to NADPH. So this is the end of this electron transport chain during light reaction, that is non-cyclic photophosphorylation. This entails a convergence of electron transport chain with the protons in the stroma, as this FNR catalyzes oxidation of 2 reduced ferredoxin molecules to reduce 1 NADP positive, and this involves transfer of 2 electrons and 1 proton from the stroma. So this The next question is, bronzing of leaves is typical caused by deficiencies of, our options are, that is molybdenum, iron, chlorine or nitrogen. So its right answer is nitrogen. So these are different minerals and their deficiency symptoms and their toxicity symptoms when they are present in excess. So firstly, nitrogen. So its deficiency causes light green leaf color and plant color up with the old ages, leaves turning yellow and leaves that will eventually turns brown and will die. So plant growth is slow When the soil is deficient in nitrogen, plants will be stunted and will be mature early. Then its excess, so plant is dark green in color, and the growth will be succulent, susceptible to subjected to diseases and insect infestation, and subjected to drought stress. Plant will easily lodge. And blossom abortion means flowers will abort. Lack of fruit set will occur. Next is ammonium toxicity. So it is caused by the application of excessive fertilizers, and it shows some toxicity symptoms like carbohydrate depletion and reduced growth. Lesions may appear on stem, and downward cupping of leaves, decay of the connective tissue at the base, and wilting of plant under moisture stress. Next, it also causes the magnesium deficiency. Excess of ammonium causes a magnesium deficiency. Next element is phosphorus. Its deficiency results in slow growth and plants become stunted, and the older leaves will have purple coloration, particularly on the underside. And its excess have no direct effect on the plant but may show visual deficiencies of zinc, iron, manganese, and high phosphorus may also interfere with the normal calcium nutrition, so it can cause calcium deficiency. Next element is potassium. So its deficiency can be seen on older leaves, and the edges will look burnt. A symptom known as scorching symptom appears due to deficiency of potassium. Plants will easily lodge and be sensitive to disease infestation. Fruits and seed production will be impaired and poor quality. Its excess inhibits the typical magnesium intake and possibly causes calcium deficiency due to cation imbalance. Next, we have the important element, that is calcium. Its deficiency can be seen in the growing tips of root and leaves, and turn brown and die. The edges of leaf will look ragged as the edges emerging leaves stick together, and fruit quality will be affected with the occurrence of blossom and rot of fruits. Its excess exhibit typical magnesium deficiency symptoms, and when in high excess, potassium deficiency may also occur. Next is magnesium. Its deficiency is observed in older leaves. They will turn yellow in color and causes intervenal chlorosis, that is yellowing between the veins. Plant growth will be slow, and some plants may easily infested by diseases. Its excess causes cation imbalance, showing sign of either calcium or potassium deficiency. Next is sulfur. So its deficiency is seen in the form of light green color of the entire plant, with the older leaves being light green to yellow in color as deficiency intensifies. Next, its excess causes premature senescence of leaves. Next is boron. It is very important for pollen germination. So its deficiency causes abnormal development of growing points, that is meristematic tissue, apical portions, and fruits aborts. For some grains and fruit crops, yield and quality is significantly reduced. Then its excess causes dieback disease of leaf tips. Next comes chlorine. Its deficiency can be seen in younger leaves, having chlorotic patches on the leaves, and plant will easily wilt. For wheat, a plant disease will infest the plant when chlorine is deficient. Its excess causes premature yellowing of lower leaves and burning of the leaves margin and tips. Leaf abscission will occur, and the plant will easily wilt when it is present in excess amount in the soil. Next element is copper. Its deficiency causes slow growth in plants with distortion of young leaves and death of growing points, and its excess interfere with iron absorption, so it may cause deficiency of iron and result in slow growth. Root may be stunted. Next important element is iron. So its deficiency causes intervenal chlorosis, slow growth. When suya deficiency, so the entire plant may be light green in color. Its excess causes bronzing of leaf. and the typical symptom frequently occurring with rice. Next is manganese. So it is very important component of OEC, that is oxygen evolving complex, which is associated with photosystem 2. So water splitting is done by this mineral. So this is a part of this OEC, that is oxygen evolving complex. Its deficiency causes intermittent chlorosis in young leaves, while the leaves and plant remain generally green in color. When severe deficiency occurs, the plant will be stunted. Excess of manganese in the older leaves will show brown spot surrounded by chlorotic zone and circle. Chlorosis means yellowing off leaves simply. Necrosis means appearance of dark spots due to death of tissue. Next is molybdenum, which is very important for nitrogen fixation. It is involved in nitrogen fixation. So its deficiency will frequently appear similar to nitrogen deficiency because it is involved associated with nitrogen fixation. So older and middle leaves becomes chlorotic first, and in some instances leaf margins are rolled and growth and flower formation is restricted. That ultimately leads to loss of yield. Its excess is of not common occurrence. Next is zinc. Its deficiency on upper leaves will show interveinal chlorosis and eventual wilting of effective leaves. Leaves may be smaller and distorted with rosette form, and its excess in. The next question: the transfer of sugars from mesophyll cells to sieve tube elements in the leaf is called as. Our options are phloem loading, phloem unloading, translocation of photosynthates, or both A and C. So its right answer is phloem loading. The transfer of sugars from mesophyll cells to sieve tube element in the leaf is called phloem loading. So firstly, this food is synthesized in the mesophyll cell, then it is transferred to the sieve tubes, and after that it will move downwards to the sink. So the transfer of sugars from mesophyll cells to sieve tube elements of the phloem in the leaf is called phloem loading. On the other hand, the transfer of sugars from sieve tube elements to receiver cell of the consumption end, that is sink organ, is called phloem unloading. So here I will discuss this mass flow hypothesis of Munch, which was given in 1930. Firstly, water absorption from the nearby cell, increasing hydrostatic pressure in the mesophyll cell. So there is increased hydrostatic pressure in the mesophyll cell. Then after that, the phloem loading occurs, that is transfer of this food material from mesophyll to phloem cell, and then the water entry from xylem to sieve tube element occurs by the osmosis. So water enters from xylem vessel to phloem, and here it increases the hydrostatic pressure again. So this increased hydrostatic pressure and difference drives solutes movement from sieve tube to sink. Because sink are deficient in food material, so it will move downward in the direction of concentration gradient. The next, phloem unloading occurs in the sink cell or sink organ. Finally, water moves back to the xylem vessel by osmosis. Here we can see here. So water moved back by the process of osmosis, and transpiration pulls up water. So this is how phloem loading or unloading takes place. Next question is: photosynthates are translocated from source to sink organ mainly in the form of. Our options are glucose, fructose, sucrose, or starch. As we know that the stored food material in case of plant is starch, while in case of animal it is glycogen. So the mobile form of food in plant is sucrose. So this is the form which is translocated from source to sink. So this is the source, and from this to sink it is transported in the form of sucrose. So glucose is produced as a result of photosynthesis but is converted into sucrose. So sucrose is transported from source to sink organ with the help of phloem tissue, that is sieve tube cells or sieve tube elements. So the correct answer is sucrose. As we know that this sucrose is a disaccharide, it consists of a glucose and one fructose molecule, and they are joined by this bond which is known as the glycosidic bond. And these glucose and fructose forms are derived from two chemical compounds which are known as pyrene. Pyrene is equivalent to this glucose, and furan form is the fructose form. Next question is: photorespiration is inhibited by. Our options are 2,4-dinitrophenone, alpha hydroxysulfonates, CMU or DCMU, and atrazine. So photosynthesis is inhibited by this one, I'm not sure about this photorespiration. So CMU and DCMU are most probably this answer of this question. So the herbicide, that is dichlorophenyl dimethylurea, that is DCMU, and chlorophenyl dimethylurea, that is CMU, are also photosynthetic inhibitors. Basically what this DCMU does, it is a very specific and sensitive inhibitor of the photosynthesis. It basically blocks the plastoquinone binding site of the photosystem 2. It interferes the activity of photosystem 2. This interrupts the photosynthetic electron ट्रांसपोर्ट चेन इन द फोटोसिंथेसिस विच इज अ पार्ट ऑफ लाइट रिएक्शन. तो लाइट रिएक्शन को ड्यूरिंग फोटोसिंथेसिस ये ब्लॉक करता है एंड द प्लांट डाई बिकॉज ऑफ deficiency of food, and thus reduces the ability of the plant to turn light energy into chemical energy. Means basically it interferes the process of photosynthesis, so plant is unable to synthesize food. So this transfer of electrons between this photosystem 2 and this plastoquinone, this is inhibited by this DCMO. So these are basically inhibitors of photosynthesis, and they are used as a herbicide to kill the plants. So this was all about for our today's discussion about these questions. Hope you will get some idea from this presentation. If you have any questions, queries, and any suggestions, you can give it in the comment section. Thanks for watching. Have a nice time.